Again.
Okay, great. So I'm going to be talking today about plant defense against herbivory. This is one of my favorite topics, so I'm happy to share it with you. And when you think about the diversity of life on Earth, if you look at just the described species, there are many species that are not yet described, but if you look at those that are, you can see a substantial portion of those described species. The teal on the left are insects. And within insects themselves, there's also tremendous diversity. And not all of these insects eat plants. They interact with them in many ways, but about 60% of known insect species do eat plants. And insects arose between 400 and 500 million years ago, so around the same time as angiosperms, and they have been interacting with them for hundreds of millions of years. And if you look at the fossil record, it's really hard to get fossils that really clearly demonstrate that insects are eating plants, because both of those organisms have pretty soft tissue. It doesn't preserve well in the fossil record. But there are records of herbivory going back to the early Devonian times. So that's over 400 million years ago. And this is a more recent fossil. So this is from the early Cretaceous, so much more recent than that. But this is a beautiful set of fossils that was relatively recently found. This was just published this year. From Nebraska, and it shows pretty clearly these scale insects on a laurel plant. So when you think about the hundreds of millions of years that insects have been around and eating plants, you might wonder why more plants don't look like this. And the answer lies in evolutionary biology and the Red Queen hypothesis. So this is from Alice in Wonderland. And the Red Queen says to Alice, "It takes all the running you can do just to keep in the same place." And this kind of characterizes the co-evolutionary relationship between plants and the herbivores that eat them. The herbivores are continuously evolving new attributes that allow them to better use particular plants as host plants for food. And the plants in turn have to be continuously evolving tweaks to their defense systems that prevents them from being just completely devoured by the insect herbivores. And when we think about plant herbivore interactions, I mainly think about it in natural systems, but there's some really nice work in agricultural systems. Because they're so well studied, they can give us quite a bit of information about how insects are responding to particular types of defense. So a lot of the synthetic chemical compounds that are used in agriculture or in pest control, like control of mosquitoes to prevent malaria, things like that. A lot of those synthetic compounds are mimics of natural plant compounds. And then a lot of the organic compounds that are used in pest control are derived directly from natural plant secondary compounds. And so if you look at how quickly insects can change in response to some of those synthetic chemicals, it can provide us with just a bit of a glimpse about how quickly they might also be doing things in natural systems. So this figure shows time on the x-axis in decades, and on the y-axis, it's the number of resistant arthropod species. And that blue line is tracking the number of resistant species. And then each of those yellow bars shows a particular chemical compound that's been used in agriculture or other purposes. And the bar starts at the date that the compound was first used. So for example, DDT at the bottom of that graph, we started using it around 1940. And then that date in parentheses after the compound name, 1946 for DDT, is the date which insect resistance to that compound was first observed. So you can see this is really rapid. DDT, we started using it around 1940, and just 6 years later, we first saw arthropod resistance to it. And it's a really similar pattern for those other compounds as well. And so it's clear from looking at non-natural systems that insects can really rapidly evolve resistance to commonly used insecticides and chemical compounds. And part of this is because of their short generation time, especially relative to many natural plant populations. Insects can go through multiple generations in the time that even an herbaceous plant, if it's a perennial, just goes through one generation. And so that really gives insects an evolutionary advantage. And it's a little bit complicated for plants to be able to keep up with the insects. But they do a pretty good job in most cases. And that is because they have many different types of defense that they use. So the first two, physical and phytochemical resistance, are both direct resistance traits. And these are decreasing the quality of food for the insect herbivores, or decreasing the quality of shelter for the herbivores on the plant. And I'll go over each of these in a bit more detail after this slide. In addition to those forms of direct resistance, plants also have different forms of indirect resistance. So there's kind of a filter in between what the plant does and the effect that it has on the insect. And plant volatile communication is one type of indirect resistance that I'll talk about a little bit more later on in the talk. So starting with physical resistance, these are traits that interfere with the ability of the herbivore to move around on the surface of the plant or to establish a good place to feed. So things like thorns, spines, hairs, or also called trichomes, are all great physical defenses for plants. And in monkey flower, so this is one of the species that I study that is native in Arizona. And on the left is a picture of a monkey flower population that's on the San Francisco peaks. Monkey flowers have these glandular trichomes that are pictured on the right. And so these are leaf hairs that are coming out from the surface of the plant. And in some monkey flowers, the trichomes also have this glandular exudate on the tips. And glandular exudate can contain chemical compounds. In monkey flowers, it doesn't seem to. It's very sticky. It contains polysaccharide sugars. So if you think about the kind of sliminess when you cook okra, that's also polysaccharides. It's a similar situation to that in some populations of monkey flower. And those trichomes in monkey flower can interfere with the ability of small herbivores to be able to eat the foliar tissue as rapidly as they want to. So this is a video of a buckeye caterpillar eating a monkey flower plant. And you can see that it is eating the trichomes off the surface of the plant. It eats them off of a particular patch of the plant before it eats the leaf itself. And the trichomes, they do have those sugars in the exudate at the tips of the trichomes, but in relatively small amounts. And the trichomes are have a lot of structural substances in them, lignin and cellulose and things like that. So they don't have great nutritional quality. And so in the caterpillars having to eat the trichomes before it consumes the foliar tissue, it is slowing down the amount of nutritional material that the caterpillars can eat, and ultimately will slow down the caterpillar growth a little bit. So trichomes can be very effective physical defenses. Phytochemical defenses, these are also called secondary metabolites or secondary compounds. And these are chemical compounds that are present in the plant. And they're called secondary metabolites to contrast them with primary metabolites. And the primary metabolites in plants are metabolites that are involved in kind of the daily functioning of the plant. So in photosynthesis and respiration, in growth, other signaling, things like that. Those are primary metabolites. And so these secondary metabolites are thought to have evolved primarily, their primary function is in defense of herbivory and to help protect them from pathogens as well. So plant secondary metabolites, there are hundreds of thousands of compounds that exist, and they have many different modes of action. They can be digestibility reducers, taste deterrents. Many of them are toxic and are neurological disruptors for anything eating the plants. The categories that you see of secondary metabolites are usually consistent within a plant family. So if there's a particular class of chemical compound, you won't necessarily see each of the same compounds across a plant family, but you will see compounds that are within a particular plant class kind of distributed across a particular plant family. And humans, we have kind of co-opted those many of those secondary compounds, and we use them in food and in medicine. And so if you think about things like cabbage, kale, broccoli, cauliflower in this picture, this is actually an interesting example of plant breeding as well, because each of those, they're closely related to one another, and they're all actually derived from this brassica plant through selective breeding for different attributes. So cabbage was selected for increased terminal buds, kohlrabi for the stem, etc. But when you think about these plants, they all have kind of a distinctive taste that they taste different, but there's something about them that makes them taste somewhat similar to one another as well. And that is actually the secondary compounds. Glucosinolates are really common in plants in this genus. And they give it that distinctive taste. And they also give these vegetables nutritive qualities that we as humans enjoy when we consume them. So I apologize for the small font on this one, but this is just some examples of plant secondary metabolites that are used in medicine. This is just a few of the long list. And so this table shows different plant species, particular secondary metabolites that occur in each of those species, their mode of action, and then the properties, the medicinal properties that humans use them for. So this just maybe 15 examples shows secondary metabolites that we use for things like tumor therapy, as muscle relaxants, as stimulants for treatment of gout or Alzheimer's. And so there's just a really broad diversity of functions that humans have co-opted these plant defenses and use them for. So this slide shows some just diversity of secondary metabolites. These are all alkaloids, which is a particular class of chemical compound. And it shows the different plant species that they occur in. And so this just shows a very, very small amount of the diversity of secondary compounds. And you can see it includes things like morphine, nicotine, and caffeine. And so those are all secondary metabolites that humans use for just enjoyment in coffee, or for medicinal purposes or other purposes. But these are also compounds that have really strong effects on insects that eat the plants that contain them. Caffeine and nicotine are neurodisruptors in insects. And so humans can consume them in relatively high amounts without immediate harm. But the same is not true for many of the insect herbivores that eat these plants. Another example that you all are likely familiar with is in stinging nettle. And stinging nettle actually has this kind of beautiful combination of physical and phytochemical defenses that it uses to protect itself. And so if you look at the nettles in the picture at the bottom, these are actually trichomes, and they are hollow trichomes that have secondary compounds within them. And when you brush against the tip of the trichomes, the tip breaks off really easily. And those trichomes essentially act as little miniature syringes. They have histamine and a number of other secondary metabolites in them that then causes us to feel the pain that we feel when we brush against the stinging nettle. Poison ivy is another plant where we relatively often feel the effects of the secondary metabolites in the plant. So the main secondary compound that we experience is this urushiol. And this is also found in mango skin and also cashews. Those are all in the same plant family as one another, and they share this secondary metabolite. The interesting, and so you might hear about people who are allergic to mangoes or cashews. I think it's relatively common, really. The interesting thing about poison ivy is that it is really just humans and other primates that show a response to the secondary metabolite. Insects and deer, other browsers can eat poison ivy without having the same reaction that humans do. And this is because this compound, when we brush against it, it gets on the surface of our skin. And the compound binds to proteins that are in our skin cells and interferes with the way the skin cells communicate with each other. And so that itself does not cause the reaction. What causes our reaction to poison ivy is actually our immune system recognizing that something isn't quite right in those skin cells. And our reaction is actually caused by kind of overstimulation of the immune system as it responds to this perceived threat. Another really interesting system for looking at phytochemical defenses and insect herbivory is this wild parsnip system. So wild parsnip is not native to North America. It's introduced from Eurasia, but it does have some close relatives that are native. Cow parsnip is native to Arizona and other parts of the US. And wild parsnip has some pretty nasty and potent secondary compounds in it. And it really only has a few herbivores that are able to consume the plant. And those are parsnip webworms and black swallowtails are two of the most common. And wild parsnip has these secondary metabolites called franocumarins in it. And these cause contact phytophotodermatitis when we get them on our skin. So this is a picture of someone who's gotten it. You can see it actually looks a lot like a poison ivy rash. And I've gotten it. It feels a lot like a poison ivy rash as well. But it has a very different mode of action from the secondary metabolites in poison ivy. So what happens with these furanocoumarins is that if you touch the surface of the leaf or even the flowers, they have the furanocoumarins in them. You get some of the compounds on your skin, and it's actually totally fine if your skin is if your skin is not exposed to sunlight. But if your skin is exposed to sunlight, then the UV light activates the action of some of these furanocoumarins, and that's what causes the unpleasant rash. But this system is really interesting. It's well studied, and so we know a lot about what the insects do in response and how they're able to deal with these phytochemicals. And one thing that the insects do to avoid the effects of the furanocoumarins is they roll a leaf around them, and then they eat the leaf tissue from the inside out. And so they are not exposed to any UV light in that situation. And so they're able to consume the plant material that contains the furanocoumarins without it actually being activated and being toxic for those herbivores. Those are actually also specialist herbivores that eat wild parsnip. So in addition to these behavioral adaptations, they also have some enzymes that help them to detoxify those secondary metabolites. Another plant insect system that most people are familiar with is monarchs and milkweed. And milkweed contains pretty potent physical defenses and also secondary compounds. And the primary physical defense is latex. So you can see in the left picture, the white substance that is kind of surrounding that monarch larvae. And so this is a really sticky substance that as soon as part of the plant tissue is damaged, the latex kind of rushes out and floods that particular area. Milkweed also contains cardenolides, and these interfere with the sodium pump that all animals have, humans, insects, etc. And it causes the heart to beat stronger and faster, and often results in cardiac arrest if you have too great a quantity of these cardenolides. And so the monarchs have developed multiple different ways of dealing with the different milkweed defenses. Monarchs are also specialists on milkweed. There aren't that many herbivores that can consume milkweed. And so the left picture shows one of the behavioral responses that monarchs have, and that is they do this cutting around a patch of tissue that they're going to eat. And in doing so, they're basically cutting off the flow of latex into that bit of plant tissue in the middle of the cutout circle. So they'll cut a circle out, kind of stop the latex at the margins, and then they're able to eat the plant tissue that's within that circle. They also have a few mutations that studies have shown just in the past few years that monarchs have developed, and the mutations are in the structure of the sodium pump. And so those mutations allow the cardenolides to interact with the sodium pump without having the same effects that they do in other animals. And one of the benefits of being a specialist herbivore and being able to circumvent or partially avoid some of the plant defenses that other herbivores can't, is that you have a lot less competition for your primary source of food. So I said that not many herbivores other than monarchs can eat milkweed, but one other herbivore that can is this milkweed bug here. And it sequesters cardenolides. The monarchs do as well. And so basically the sequestration, the insects are consuming the plant material, and then those secondary metabolites are going out from the digestive tract and into the body of the insect. And so if a predator were to try to eat one of these insects, it's going to taste terrible, but the predator also has to deal with some of the toxic effects of those secondary metabolites because they have been moved into the body of the insect. And like the monarchs, the milkweed bugs also have this warning coloration. And it's these bright patterns and colors that warn predators that it would be a bad idea to try to eat it. All right, this is another example of a combination of physical and phytochemical defenses. So you might be seeing a trend here. Most plants aren't able to just rely on one particular type of defense, again, to try to keep up with the insects that are consuming them and the adaptations that the insects have. Most plants have some combination of physical and phytochemical defenses. And within the phytochemical defenses, they usually contain multiple secondary metabolites. And so all of that is necessary just to try to keep one step ahead of their insect herbivores. So this example is in Bursera plants. So these are flowering shrubs and trees that grow in Central America. And the herbivore here is another specialist. It's a chrysomelid beetle larvae shown on the right. And the defenses of these plants are really, really interesting. So they are terpene containing resins. So terpenes are a particular class of secondary metabolites. And so there are these resins containing the secondary metabolites that are stored under pressure in canals in the leaves and stems of these plants. And when the canals are punctured by an insect feeding on them, for example, you can see in the left picture that there's something kind of squirting out of that piece of leaf. And so these high pressure resins are propelled out when the canals are punctured. And the resins are toxic because of the terpenes, and they also solidify when they're exposed to air. And so actually some small insects that might eat these plants can become basically entombed inside these resins. And so this particular herbivore, again, it's a specialist. And so it has evolved mechanisms to be able to partially circumvent the defenses in these plants. And so what this beetle larvae does is you can see underneath the body of the larvae, there are those little kind of dark spots in the central vein of that leaf. And so it is basically cutting through the canal, or one of the canals that carries those resins to the tip of the leaf. And so in doing so, it's decreasing the pressure and preventing that kind of high pressure propulsion when it chews on the tip of the leaf. And so it is similar to the monarch, where it cuts off the flow of latex. This herbivore is preventing some of the physical attributes of that defense from being able to affect its feeding. All right, so that was kind of an overview of some of the physical and phytochemical resistance mechanisms that plants have. And again, those are all ways of directly affecting the herbivores that are eating the plants. And so I wanted to talk a little bit now about indirect defenses. So again, when an herbivore feeds on a plant, it experiences these direct defenses that the plant has. But the plant also has other forms of defense against herbivory. And one of the main forms of indirect defenses is in the plant attracting predators of the herbivores. So if an herbivore is chewing on a plant, the plant has many different ways of attracting predators who then eat the herbivores. And that in turn reduces the amount of herbivory that the plant is experiencing. And one relatively common form of indirect offense is extrafloral nectaries. These are extra floral because they are occurring outside the flower. They're often outside and kind of near the base of a flower. These pictures show extrafloral nectaries in barrel cactus on the left and viburnum plants. And these extrafloral nectaries contain a lot of sugar, as you might imagine, but they also carry many other really nutritious things for insects, amino acids, lipids, and things like that. And they occur in at least 4,000 species. These are just the plant species that these have been characterized in over 100 plant families in the world. So they are relatively common. And with the kind of reward that they offer, they attract a lot of different predatory arthropods. And so those include things like ants, as shown here, and also wasps and spiders. And an interesting case of extrafloral nectaries occurs in sunflowers. And so any of you that have sunflowers growing here in Arizona, you may have noticed that your sunflower plants have a lot of ants kind of running up and down all over the stems and leaves. And this is actually for two reasons in sunflowers. One, they do contain extrafloral nectaries, so those are attracting the ants. But you might also notice that your sunflower plants often have a lot of aphids on them. And there is this really interesting mutualistic interaction between ants and aphids. So the aphids are exuding honeydew from their bodies, which is a very sugary substance. And so ants will often tend to and protect the aphids in return for that honeydew reward that they get for doing so. So sometimes ants will eat aphids, but they don't eat very many of them relative to how many aphids there are. And so that mutualistic relationship between ants and aphids is not great for the plant because those aphids are feeding on plants, usually in really high densities. And so there's some evidence that these extrafloral nectaries actually evolved in order to help distract ants from tending to the aphids and offer them this kind of alternative reward for protecting the plant. And so while the ants often don't eat aphids, and that's more of a mutualistic relationship, the ants are actually very aggressive, no matter what species it is that's attracted to these extrafloral nectaries. And so they will deter most other herbivores from feeding on a plant. So caterpillars, beetles, most other herbivores besides aphids will be deterred from feeding on a plant by these ants, which are attracted by the extrafloral nectaries. All right, so plants have all these different types of defenses: direct, indirect, physical, phytochemical, but plants are also not static individuals. They can't move usually, but there's a lot going on within a plant. And so plants are able to induce or upregulate their levels of defenses while they're being eaten or after herbivory. And they can do that within a particular plant itself. Or they also use different methods of plant-plant communication. And they can communicate with plants that are in the immediate vicinity and send signals depending on the amount or type of herbivory that they are receiving. So you probably have seen things about this in the news in the last 5 years. Talking trees is a really kind of common blurb that is in the news about plant-plant communication. And so what happens in this particular type of defense, so this is really an indirect type of defense, is that a plant, when it's being eaten by, say, a caterpillar, will start to emit these volatile organic compounds, or VOCs. And those volatile organic compounds, there are, like secondary metabolites in plants, there are hundreds of thousands of forms of these volatile organic compounds, and they have different functions. And one of the functions of them is that they attract predators and parasitoids of the herbivores. So the predators are directly, they're recruited to the plants by these volatile emissions, and they're going to eat the insect herbivores. Parasitoids are a little different. These are things like parasitoid wasps, and they are inserting their eggs into the body of the herbivores. And when the larvae of the parasitoids start to emerge from those eggs, the larvae are using the body of that herbivore as a food source. So they're basically consuming the herbivore from the inside out. And so that's a bit more of a delayed mechanism to get rid of herbivores, but it does eventually result in herbivore death through parasitoids. So the volatile organic compounds are attracting predators and parasitoids of the herbivores, and they are also signaling to other plants. And so if there's a plant that is getting really heavy herbivory, it's going to have volatile emissions that are detected by plants surrounding it. And it can cue those plants to upregulate their own levels of defense, even in the absence of getting any herbivory themselves. And so it's called priming. They're priming themselves to be ready and a bit more defended when that herbivory does actually come to them. And this communication doesn't only happen above ground. This figure shows that is also occurring between root systems of different plants. And there's also communication through these volatiles from the roots to the above ground tissue within a plant. So within a plant, the communication is going through the plant vascular network, but it's also going through the air, in part through these volatile organic compounds. All right, so I talked earlier a bit about how different herbivores are able to avoid or ameliorate the effects of particular defense compounds. And so in one last example, I wanted to talk a little bit about galls. And these are these really often beautiful, charismatic structures that form on plants. And these are caused by a really wide variety of species, gall midges and gall flies, gall wasps, different mites, aphids, sawflies, and a few moths and beetles can all cause these galls to form on plants. And the interesting thing is that each insect species produces or causes the plant to produce a unique gall. And what happens with gall formation is the insect starts to feed on a particular area of a plant, and there are cues in the saliva of the insect that alter the plant growth hormone in the tissue that's surrounding where the insect is feeding. And that increase in plant growth hormones results in abnormal cell growth in the area right around where the insect is feeding. And that abnormal cell growth eventually causes the cells to form into this gall shape around the body of the insect herbivore. And the galls are lined with nutritive tissue for the insects. And so the insect herbivore is protected by that gall structure from being preyed upon. And it has really nutritious material from the plant to eat. The insects in these galls can manipulate a number of different parts of the plant system. So one of the things that they can do is actually alter the source-sink relationship within the plant and even cause an extra flow of nutrients to be going into the galls themselves. Another really interesting thing that just a few galling wasps can do is they can actually cause the plant host to produce a sugary substance that's like honeydew right near the formation of the gall. And that honeydew, like the extrafloral nectaries, will attract ants. And the ants are acting as an additional layer of protection for the herbivore against predators that may try to work around the gall. So these gall-forming insects are really fascinating. They can manipulate the plant in many ways to their own benefit. And this is really kind of a growing field of study in plant biology, is looking at what the pathways are that lead to gall formation and what exactly is going on in the insect-plant interaction to cause the formation of these galls. All right, so that was kind of my overview of plant defenses. I hope that I've given you kind of a snapshot of many of the different kinds of defenses that plants use against insect herbivores, and also a little bit of information about some of the many different ways that insects can or have evolved to be able to deal with some of those plant defenses and to better feed on plants. So to wrap up here, I wanted to just give a brief overview of my research system and some of the work that I do. So I work a lot with yellow monkey flower. So this is occurring around us in Arizona. It's native to Western North America. I also work with the red monkey flower that occurs in northern, well, actually all of Arizona as well, but I'll focus just on the next few slides here for this talk on my work in yellow monkey flower. So this species is actually a model system for a number of scientists that are interested in integrating genetics and different aspects of evolutionary ecology. And this is for a variety of reasons listed here. But one of the reasons why I really love this species is because of its great diversity of habitats. And so the pictures on the bottom are from, are of a number of monkey flower populations. On the left, we have alpine populations in the Cascades in Central Oregon. And so these are really tiny annuals that usually produce just one pair of true leaves and one flower. And they're growing in the water and the slope from snow melt. And so the snow melts, the plants, have a really short window to grow and potentially be pollinated and set seed before the water in that slope disappears and the plant season is over. They grow in sand dunes along the coast of Oregon, in cliff sides and ditches and other riparian areas. And in Arizona, we don't have quite as much surface water as some areas of along the West Coast. And so in Arizona, these are mainly occurring in springs and seeps, and also some of our riparian areas. And we work with a really large number of natural populations. So we've collected from a lot of the native range of these monkey flowers. And also Mike Rotter, a previous PhD student in the lab, was interested in non-native monkey flowers. So he collected also in their non-native range in the eastern part of North America, and also in the United Kingdom. And so each of these dots actually in the Western US represents multiple collections within kind of a small area. And so what we do with all of those plants derived from all these different natural populations is we are really interested in the defenses of these monkey flowers and how they affect plant herbivore interactions. I'm interested in the genetic basis of the defense, so the genes that are underlying production of the defenses. And so I talked a little bit about trichomes before and how they were reducing the amount that particular caterpillar larvae was able to feed on the plant. Monkey flowers also have these suites of phytochemical defenses that they produce. And in addition to kind of looking at the level of the plant and the defense compounds and interactions with individual herbivores, we study things at more of a landscape level. So on the left is just some natural populations that we looked at the chemistry of when these plants were grown in a common garden in the greenhouse. And the size of the pie represents the concentrations of the phytochemicals in the plant. So the bigger the pie, the higher the concentration of these defensive compounds the plants in that population have. And then the different colors in the pie represent different individual phytochemicals. And so you can see that the concentrations of their phytochemical defenses and also the composition of those phytochemical arsenals is changing across the landscape. And so we're really interested in the biotic factors, such as herbivores, and also abiotic factors like climate that affect the evolution of these defenses in these natural populations. And then on the right side of this slide is phylogeny of some of the closest relatives of yellow monkey flower. And I'm also interested in macroevolution of defenses. So how the defenses have changed as you move across different species and within the genus of this group of plants. All right, and so in addition to those kind of larger scale issues, we do a lot of work looking at the interactions of particular herbivores with the physical and chemical defenses of the plant. So this is a buckeye larvae that is hanging out on a monkey flower plant that's in Oak Creek Canyon. So we have these beautiful places to work really close to Flagstaff. And the interesting thing about these buckeyes is that they are also specialists, like a lot of the herbivores that I've talked about in previous slides in this talk. And so the buckeyes we have found are actually sequestering some of the chemicals that monkey flowers contain. So the structures that I showed 2 slides ago are these kind of big chemical compounds that contain sugars and many other things. And the buckeyes are actually breaking those chemicals into pieces when they consume the plant tissue with them. And then they are consuming the sugars, and then the different pieces of the phytochemical compounds, they're moving them out from their guts into different areas of their body to protect them from predation. And so you can see this buckeye here doesn't seem particularly concerned with whether it's seen by predators or not. You see this a lot. They climb out to the very tips, and they're these big dark things against the bright yellow of the flowers. So they're very conspicuous. And so their specialization on some of the compounds in monkey flowers is allowing them to really drastically cut down on the rates that they are eaten by things like birds, or parasitoids are laying their eggs within the caterpillars. So just one last example of one of the many complicated ways that insects can evolve in response to those plant defenses. All right, and with that, I hope I have shared some of my love and fascination for all of these really complex ways that plants and their herbivores interact. This is actually just sort of glancing at the surface of many of these interactions. But I hope I've given you a number of good examples of how plant herbivore interactions work in a number of different systems. And I am happy to take any questions that you have.